The function of the forked tongue of squamates has been discussed for thousands of years, dating back to Aristotle's theory that a forked tongue would give snakes a twofold sense of taste. Centuries later, Italian astronomer Hodierna hypothesized that because snakes slither around so close to the ground, they use their forked tongues to pick particles out of their nose. A 17th century theory hypothesized that snakes use their forked tongues to catch flies out of the air. Another myth is that snakes use their tines of the tongue to sting their prey. Today, all of these hypotheses have been disproved. Kurt Schwank's 1994 article, Why Snakes Have Forked Tongues, supports the idea that forked tongues are used to instantaneously sample the chemical gradients of their surroundings, allowing them to follow scent trails to track mates and prey. The forked tongues of squamates has evolved at least 2 times, and its function has been perfected over time to enhance its survival and reproduction abilities. There are different mechanisms of animal movement, and Schwank discusses these in his article to determine the mechanism that forked tongues fall under. In Drs. Gottfried Frankl and Donald Gunn's 1961 book, The Orientation of Animals, Kinesis, Taxis, and Compass Reactions, they discuss the difference between kinesis and taxis. Kinesis and taxis are different types of movement. Kinesis is undirected, random reactions and movements of an animal due to a given stimulus, while taxis are directed movements and reactions in response to a stimulus. Frankl and Gunn go on to distinguish between the types of taxis. For the case of squamates, the 2 most important taxis to discuss are clinotaxis and tropotaxis. Both of these taxis allow an animal to assess their environment through continuous sensory assessment and inform the animal of the location and direction of a given stimulus. Clinotaxis involves a single receptor continuously sampling the environment to determine the location of a stimulus. It is characterized by wide-ranging movements, often involving side-to-side head motions, as the animal collects and compares successive stimulus intensities until they can determine the direction of a stimulus. Tropotaxis involves 2 receptors, or sense organs, simultaneously comparing stimuli on 2 sides of the body, allowing them to quickly identify the location and direction of a stimulus. This is typically characterized by a straight path of movement towards or away from a stimulus source. The forked tongue of a squamate is hypothesized to function under a tropotactic mechanism. Once a chemical source is encountered, the 2 tines of the tongue act as 2 receptors, simultaneously detecting the chemical zone and delivering the stimuli to the vomeronasal organs, or the VNO. This allows them to locate the stimulus signal in space and to follow a stimulus trail, such as a pheromone trail, with little to no divergence from that trail. The depth of the tongue fork is directly related to the ability to follow a scent trail. The deeper the fork, the greater ability to follow a stimulus. Observations of many squamates with forked tongues has also provided evidence that the wider the tongue fork is spread while trail following will allow for greater ability to sample a chemical gradient, because there is greater distance between the 2 sampling points. In a 1930s experiment by German biologist Hermann Kamann, he tested this theory by removing snakes' forked tongues. This resulted in a lessened or fully eliminated ability for the snakes to follow a scent trail, but did not prevent the snake from delivering a stimulus to the VNO. Another experiment by Neil Ford in 1986 observed male garter snakes following female pheromone trails, and also suggested that forked tongues function under tropotaxis. Ford observed that the tongue tines were widely laterally spread while following the trail, and if one tongue tip exceeded the pheromone trail during a tongue flick, it was able to quickly return to the trail before the next tongue flick. The snake's trail following abilities were accurate and followed a straight path.
With the specificity of chemosensory in snakes and lizard tongues, there is also a correlation between tongue form and foraging modes. In Schwenn's study, the researchers were curious if the degree of forkedness of the tongues affected the foraging modes, or if there was a correlation between the 2. In this study, they gathered data from various families within the Squamata taxa and found the average tongue tip bifurcation, and they also gathered data on their foraging modes. They looked at species that were either sit-and-wait foragers or wide foragers, and what they found was that there was a positive correlation between the presence of a forked tongue and wide foraging. So those who have to travel a longer distance benefit more from a forked tongue due to its heightened chemosensory abilities. The correlation between tongue form and foraging modes can be seen in the differences between lacertids and varanids. Lacertids, which are pictured on the right, hunt prey in relatively small ranges. Varanids, on the other hand, usually travel long distances to hunt their prey. Since varanids follow a wide foraging mode, they have a dramatically forked tongue, while lacertids do not. Their dramatically forked tongue is extremely beneficial to the varanid because it helps in chemosensoring their prey. While forked tongues are extremely beneficial for chemosensory, they also have negative consequences. In Cooper's study, they specifically investigated this topic. They found that lizards and snakes who have extremely forked tongues fail in other tongue functions. For example, they're unable to transport food or they have trouble with intraoral manipulation. This especially affects varanids who have deeply forked tongues and they are unable to do these things. This research was especially interesting because it shows that the forked tongue does have some benefits, but also has some drawbacks as well. Since tongue shape does affect foraging modes, it is to be expected that evolution and forked tongues have a unique interaction. The snakes and lizards that we most commonly think of today fall under the Squamata taxa. It is one of the most diverse radiations of terrestrial vertebrates on the planet with over 9,400 species. And with these species, they all span every continent except for Antarctica. With this level of diversity and expansion, the evolution of the Squamata taxa is important to understand forked tongues. Here is a phylogenetic tree of the Squamata taxa and a few of its families. Unfortunately, we do not have the entire Squamata taxa resolved, but we do know that the forked tongue has evolved independently at least twice within it. It has evolved at least once in the major lineages of Squamata into Angromorpha and Scenomorpha, and these can be seen on the right side of the phylogenetic tree. And then within each of these major lineages, forked tongues evolved once more. And then the divergences in evolution can be seen in the organisms. For example, iguanas, who are not a part of the first initial divergence of forked tongues, they can be seen on the left side of the phylogenetic tree, and they do not tend to have forked tongues. However, on the other hand, lacertids, who were a part of the initial divergence and then once more diverged, are seen to have deeply forked tongues.